Professor, back in the 80s, you described a biological clock in fruit flies. Of course, this is not a clock made of digits and minute hands. It is made of molecule and proteins. How does it work?
Right, and of course, compared to digital clocks or mechanical clocks, it's much more ancient. This is maybe the first clock that had 24 hours connected to it. It's a clock that has evolved multiple times. For example, we think a similar clock organized to tell cyanobacteria time of day operates by different principles. But so far as we know, all of the clocks in the animal world and in the plant world, the fungal world, operate through a series of genes that produce proteins that are auto-inhibitory, meaning that they control their own level of production. And they do so in a cyclical pattern so that they're expressed once per day. They inhibit their own action once a day, and they are disinhibited once a day to produce oscillations. And because many of the components are themselves gene regulatory components, transcription factors, this opens that oscillating subsystem up for regulating broadly gene activity across the genome. So these are clocks that not only control our behavior, our sleep-wake cycle, but many other physiological attributes. They're present in us, for example, in every organ in our bodies. They're probably present in most cells, if not all cells. And they're keeping time locally. They're usually in synchrony with one another, but sometimes they get out of synchrony. Jet lag will desynchronize them, for example. So when we recover from jet lag, it's not so much getting one clock reset to the right time. You've got this collection of millions of clocks that are running at their own, with their own phase, bringing them all back to synchrony. So we use genetics to find the key components that form these clocks and cause their operation. We started this work in the 1980s in Drosophila fruit fly, which has been a great genetic model for all kinds of biology over and over and over again, even going back to the fundamentals of genetics itself. In the early 1900s, it was Drosophila that gave the first real insights into how genes are organized on chromosomes, and most of the early ways that genetic function was dissected was done in Drosophila. And then eventually bacteria and bacteriophage, bacterial viruses. But Drosophila has made contributions once again to letting us step into this arena. And it turns out that the genes that we found in Drosophila that cause this cycling intracellular activity, the same genes are found in us, and most animals have the same collection of genes. There are some modifications, some differences, but the basic principles for how they work are the same from fruit flies to humans.
And as you said, this clock is involved in all sorts of gene expression regulation in every organ, basically. And the most famous, we can say, cycle that they regulate is the wake-sleep cycle, but they are involved in a lot of other functions.
Yeah.
When these clocks are alterated, there are some risks.
If we fly, for example, from here to Tokyo, then everything reinforces being on a new time frame. If we have a work schedule in New York, let's say, or Italy, that is more appropriate for the Far East, say Tokyo, we have some environmental reinforcement of that new time zone, just through the lighting at the working place, other environmental effects, but the rest of the environment is, of course, still on local time. And so there's a mixture of signals that can cause different responses depending on the tissue. We know, for example, that that the clocks are very important for the way our liver functions, for the way our pancreas functions. If you knock out the molecular mechanism for these clocks just in the pancreas, for example, in a mouse, you'll produce a diabetic mouse, even if clocks in the rest of the body are fine. Those clocks in the rest of the body are not sufficient to give the kinds of directions to get the pancreas to do everything on time, evidently. And the same would go, you'd have another set of problems if you did the same thing in the liver, for example, or the brain.
You're working recently at delayed sleep phase disorder, which is a disorder quite common in the US. Up to 5% of people are affected.
Yeah.
And in modern society, maybe this is something spreading, right?
Right, and that 5% figure is, again, a rough estimate that have been made. We don't know how much of that might be heritable and how much of it might be response to environment. This is a first indication that a big part of it can be genetic. So the genetic change that we've been studying is present in about 1% of the population. So that's 1% out of 5%. That's 20% of all the DSPD with one mutation. So it may be that we can continue to look for these kinds of disorders and find an even higher level of genetic contribution to the total. This is, we've talked for a very long time about sleep preferences differing from, individual has a sleep preference that differs from another individual. In this case, it's not really a sleep preference. It's a hardwired reflection of the biology of the individual. And that makes it very important to understand in considerable detail.
Professor, when you received the phone call from the Royal Swedish Academy of Sciences, did that affected your sleep-wake cycle?
Yeah, it was. Well, we don't have a phone in the bedroom. Our landline telephone is out in the kitchen, plugged into a recorder, and we assume that anybody that really wants to get a hold of us is going to call a cell phone, but we didn't have it plugged in. And I did get a call that morning from on my cell phone. We keep the cell phone in the room, and it was someone at the university calling me to tell me I need to answer the landline because the Karolinska was trying to get a hold of me. And it was a very interesting, exciting morning. I in addition to having a laboratory at Rockefeller, I'm also essentially the provost at the university. And so my first thought when the phone rang was they're going to need me at the university because somebody at the university has been given a Nobel Prize. And then I realized very quickly that I was going to have to show up for two reasons. So the biggest challenge that morning was getting out of the house because the phone kept ringing. And we finally had to have a strategy where I was unavailable. My wife would answer the phone while I'd get dressed so that we could get into town. And then we just had to take it off the hook a while she's getting dressed. We did get it back on the hook. We did plug it in long enough to talk to people from Karolinska because they had called multiple times trying to get a hold of us. And never tried our cell phone. I think they have a policy of only using the landlines. But it was a busy day.
Journalists can be very annoying sometimes. Thank you very much, Professor.
You're welcome. You're welcome.